Hi there, welcome back. Professor Jared Rathel here, and you have tuned in to lecture 2.3, which is entitled Phylogenetic Classification of Living Mammals. Look, I know you guys have endured some brutally long lectures this week. Hopefully you found them entertaining, but I know they've been long. So I've purposefully pared these chapters down in the textbook. I noted this in Canvas, but for lesson 2.3, you're only going to be responsible for chapter 2, which is, of course, methods for studying mammals. I only want you to read the few pages under the section heading DNA sequences. That's within the systematic methods section. So that's all you'll be accountable for. And then further for this lecture, I'd also like you to return to chapter 3, which is entitled Phylogeny and Diversification of Mammals. And within that first section, that's entitled Relationships and Classification of Living Mammalian Orders, please read the paragraphs under two headings. The first is Simpson's Classification of Mammals, and then the few paragraphs under the section heading Insectivores. And so again, that's what you'll be responsible for, and these readings are noted, as always, in Canvas. For most of its history, mammalian biology relied on the study of whole animals or on their preserved remains from museum collections. However, as our understanding of the biochemical basis of heredity evolved across the 20th century, from understanding that DNA was the molecule of heredity to identifying the double helix structure. So as this science evolved, mammalogists have increasingly employed molecular markers to study population genetics, speciation, and phylogeny, our topic of interest today. So for example, the development of polymerase chain reaction, or PCR, in the late 1980s is going to bring DNA sequence and fragment analysis to labs of evolutionary geneticists around the world. In PCR, a target sequence of DNA is amplified, tremendously amplified, by first unraveling that double helix structure using heat. Okay, so this is called denaturation. Then we're going to anneal or fix primers to specific target regions. And then we're going to build homolog strands using a thermostable DNA polymerase like TAC, that's TAQ, which was actually isolated from thermophilic bacteria in hot springs from Yellowstone. At the completion of one cycle, the region of DNA between the primers has now been copied, and it's available to serve as a template during the second cycle. Okay, so by repeating this process again and again, the number of copies of this target sequence of DNA is going to increase geometrically. Okay, so in 30 cycles of PCR, it's sufficient to produce 1 billion copies of a single template molecule. All right, please put me on pause for 2 minutes and 37 seconds and hop on to Canvas and check out the embedded video short. It's a really nice animation of polymerase chain reaction. So please check that out now. So the sensitivity of PCR has made it possible to recover and then amplify and analyze DNA sequences from the remains of long dead, even extinct mammals, including species like the woolly mammoth, the stellar sea cow, the thylacine, which we talked about last lecture, and then our cousins, the European Neanderthals. Estimates of the divergence between the Neanderthals and Homo sapiens, our species, range from about 315,000 years ago to some 800,000 years ago. So if we take a mean somewhere around 500,000 years ago, the lineage, which is to produce the Neanderthals in Europe and Homo sapiens in Africa, splits. Between 60 and 90,000 years ago, a changing climate spurred humans to leave our African homeland. And then between 43 and 48,000 years ago, humans colonized Europe from present-day Kazakhstan, the cradle of humanity. Upon arriving in Europe, these first European humans encountered the Neanderthals, who had been there for hundreds of thousands of years. We now have evidence that those humans interbred with the Neanderthals, as approximately 2% of European and Asian genomes are actually Neanderthal genes. Africans, like the Son Bushman here, they have considerably less Neanderthal DNA, on the order of about 0.5%, and that introgression of Neanderthal DNA into the African genomes likely occurred as Europeans migrated back into Africa. Here's a great 2.5-minute synopsis from a natural history museum that's going to really better explain that interaction between Neanderthals and humans and the genetic evidence that we now have for interbreeding. Many molecular studies in mammalogy are going to focus on microsatellite markers, which are loci, remember loci are positions on chromosomes, that show these back-to-back repeats of about 2 to 6 base pairs. So, for example, here's an individual. Maybe it's a bat from Reunion Island. So this is a recently published study on the relatedness of a population of bats from Reunion Island, and they use these microsatellite markers. So maybe individuals from this bat colony show 4 of these 4 nucleotide short tandem repeats, or STRs. Here you can see GATA, GATA, right? So 4 times. Whereas individuals from these colonies show 6 of these STRs, these short tandem repeats. So great way to look at population level problems in genetics. So the reason why this works so well is because these loci are prone to these slip strand mispairings during DNA replication. So these STRs, they have a high mutation rate that's going to generate considerable variation across individuals. The most widely studied sequence in mammalogy is the mitochondrial DNA. So you remember mitochondria from your General Biology 1 class. Those are the organelles that are considered the powerhouses of the cell. So these are the organelles that do cellular respiration for the larger eukaryotic cell. That is to say, they convert glucose into ATP. The mitochondria evolved through endosymbiosis. So billions of years ago, there was an aerobic prokaryote, a bacteria, that was engulfed by a larger host cell. This larger cell, instead of consuming the bacteria, it actually incorporates it into its cellular architecture in this ancient mutualistic relationship. So that means that each one of these mitochondria is going to have its own circular DNA, the mitochondrial DNA, which looks a lot like bacterial DNA because these were once free-living bacteria. But this mitochondrial DNA is separate from the nuclear DNA. So it's really nice because it's only about 16,000 base pairs. And then further, the protein coding genes in the mitochondrial DNA, are highly conserved, meaning they don't mutate very often. That said, there are other loci in the mitochondrial DNA that evolve very rapidly. So this means mitochondrial DNA is very diverse in terms of the scale of the questions that you can ask. You can use mitochondrial DNA to ask intraspecific questions, so looking at populations within a species, all the way up to intraordinal questions. And I might point out that it was actually mitochondrial DNA analysis that first mapped out human migrations around the globe in what's called the Human Genographic Project. So looking at mitochondrial DNA, we were able to determine that Africa was the birthplace of humans. The first wave leaves Africa, travels across southern Asia, there's India, across Indonesia, and settles in Australia, becoming the Aboriginal Australians. Another wave, as we mentioned several slides ago, settles in Kazakhstan and then moves over into Europe to encounter the Neanderthals. Still another wave, waves really, spread out across Asia, eventually move across the Bering Land Bridge, populate North America, the Native Americans, and then down into South America. So these routes were all established looking at mitochondrial DNA across human species. Increasingly, evolutionary geneticists are now employing next generation sequencing. So this is a really powerful method which is going to determine millions of short, and by short I mean like 50 to 750 bases. They're going to determine millions of these sequence fragments simultaneously. These fragments are then assembled into larger sequences by linking them together using regions of overlap, using high-speed computational methods. These reconstructed genomes are then compared against a reference genome. Reference genomes are really important because they're going to make the analysis of new genomic data sets much easier and more robust. So I'm super stoked to report that there are hundreds of these reference genomes that are now openly available in open access databases like the National Institutes of Health. They have Gene Bank, and you can download and analyze this data. There's another project known as Zoonomia, and you can see all of these species for which reference genomes exist. It's a really wide variety, from carnivores like the Arctic fox to the ai, which is a lemur from Madagascar, a primate. So a wonderful variety of mammalian reference genomes that have allowed us to build the most precise phylogenetic tree of mammalian relationships to date. All right, let's conclude with a bit of history. George Gaylord Simpson was a professor of zoology at Columbia and a curator at the American Museum of Natural History. He was a prominent evolutionary biologist. He contributed to the great synthesis in biology. He was a paleontologist and a taxonomist. In fact, George Gaylord Simpson, he really laid the taxonomic foundation for our modern science of mammalogy with his landmark publication entitled The Principles of Classification and a Classification Classification of Mammals, which was published in 1945. As you may recognize at right, many of the orders that George Gaylord Simpson described are still in use today. So he recognized the order Monotremata, the marsupials, the primates, the chiroptera, rodents, lagomorphs, carnivores, so on and so forth. So these orders have survived decades of subsequent research. So George Gaylord Simpson, he didn't have next generation sequencers, okay? So he realized at the time that some of his orders, like the order Insectivora here, were, and I quote, something of a scrap basket for small animals of a generally primitive character that are not clearly referable to some more distinctive order. So, in short, he's going to lump the hedgehogs, the shrews, the tenrecs, the golden moles. He's going to put them all in this catch-all order that he calls the Insectivora. Using modern molecular methods, phylogenetic systematists now split the Insectivora into the orders Macroscelidae, which includes the elephant shrews, the order Afrosauricidae, which includes the tenrecs, shrew tenrecs, and otter shrews. We'll be covering both of those orders in our next lecture, so that's why we're covering classification now, as well as the Eulapotphyla, which is the solenodons, true shrews, moles, hedgehogs. We'll cover them in week 5, and then the order Scandentia, which are the tree shrews, and we'll cover Scandentia in week 4. This represents the best phylogenetic tree of mammalian orders that we have to date. So this has been presented before. It's in Chapter 3, the Phylogeny and Diversification of Mammals. This is figure 3.1. But this is going to map out those phylogenetic relationships. And remember, sister taxa, they are most closely related to each other. So the Macroscelidae and the Afrosauricidae, they are what we would consider sister taxa because they share a most recent common ancestor here, some 75 million years ago. So these are the orders that we're going to be covering in ABS 470. We've already covered a good chunk of them. Here's our monotremes and our marsupials. So you remember the New World opossums, the Dasyurids, which includes the Tasmanian devil and the thylacine. So these have already been covered, and then in our next lecture, we're going to cover these 3 orders within this superorder, the Afrotheria. So we're going to be coming back to this figure again and again because in mammalogy, in all the biological sciences, it only makes sense to see things through an evolutionary lens. With that short lecture, I told you it was short, just a handful of references cited in this lecture, and just one more lecture to go in this module before your first assessment. Thanks for your time.